Today's presentation is Introduction to Microbial Metabolism. It is aimed to assist students to be able to recall what has been discussed during the physical classroom lectures. Now, to start with, what is metabolism? Metabolism is from the Greek terminology metabolen, which means change. Therefore, microbial metabolism refers to all the chemical changes or reactions required to maintain the nutritional and functional activities of microorganisms. In other words, microbial metabolism refers to all the biochemical reactions occurring in microorganisms during growth and development. In fact, microbial metabolism can be defined as the process by which microorganisms acquire energy and materials from nutrients and utilize them for biosynthesis and other body activities. Now, generally, metabolic reactions taking place in microorganisms are categorized into two. The first category of the reactions are called catabolism. Catabolism generally refers to breaking down of complex organic substances into simpler ones. It is otherwise called degradative reactions or catabolic reactions, which involve breaking complex organic compounds into simpler substances with the release of energy. The release of energy is otherwise called exergonism. In this case, means that the reactions produce more energy than they consume. Now, catabolic reactions usually take place through the process of hydrolysis, which are reactions that use water and in which chemical bonds are broken, such as carbohydrates being broken down into glucose, protein being broken down into amino acid, lipids into fatty acid and glycerol, and so on. The energy that is released during this breaking down process is captured in the form of adenosine triphosphate, ATP, which is the energy currency of the cell. One of the common examples of catabolism is cellular respiration. In cellular respiration, glucose is oxidized. Glucose is oxidized in the presence of oxygen into carbon dioxide and water with the release of large amount of energy. In other words, catabolism can simply be defined as a process by which living organisms obtain their energy and materials. The process by which living organisms obtain energy and materials for their body activities. Now, the second category of metabolic reactions are generally referred to as anabolism, which are mainly synthetic reactions, that is, directly opposite to catabolism. Now, anabolism, which is also called synthetic reactions, involve building complex molecules from simpler molecules, and it takes place usually through dehydration or synthesis, that is removal of water from chemical bonds, and it is usually endergonic, that is reactions that produce more energy than the endergonic, which are reactions that consume more energy than they produce, such as amino acid being synthesized into proteins, simple sugars into polysaccharides, fatty acid and glycerol into lipids. The energy required for these synthetic processes is derived from ATP, which is generated during catabolic reactions. An example of anabolic process is the photosynthesis, in which carbon dioxide and water are added in the presence of sunlight energy to produce food, which is glucose, and in the process oxygen is released. That is, this reaction is directly opposite to the catabolic reactions, in which in respiration glucose is broken down in the presence of oxygen to release carbon dioxide, water, and energy. So in essence, the relationship between catabolism and anabolism is reciprocal. Catabolic reactions provide the energy and building blocks to drive anabolic reactions. In other words, catabolism provides the energy and precursors required for anabolism, while anabolism utilizes this energy and precursors to build complex molecules, as can be seen in this example. In catabolism, we have larger substances being broken down into simpler ones to release energy, and this energy that is released is now used in anabolic process to synthesize important cellular components such as macromolecules and so on. Now, each anabolic and catabolic process occur in a series of stages or reactions which are called metabolic pathway. So the next question is, what is metabolic pathway? So this takes us to the next aspect of this presentation, which is metabolic pathways and enzymes. Now, metabolic pathways are a series of enzymatically catalyzed biochemical reactions in which the product of one reaction serves as the substrate for the next reaction. And because the chemical reactions are catalyzed by enzymes, metabolic pathways present in an organism generally depend on the enzymes present in that particular organism. While the enzyme present in an organism generally depend on the genetic constituent of that organism. In simplified term, a metabolic pathway can be exemplified as follows. Now, here we have a series of reactions which start with substrate A being converted by enzyme A through dehydration process to produce a product B, which serve as a substrate for the product C under the action of the enzyme K that release carbon dioxide from product B, and the product C serve as the substrate for the next reaction catalyzed by enzyme B to produce the product D, and so on. Now, what are enzymes? Since metabolic pathways are catalyzed by enzymes, what are enzymes? Enzymes are generally biological catalysts or catalytic proteins or organic molecules that catalyze or speed up the rate of biochemical reactions by lowering the activation energy and orienting the reactant molecules to produce a desirable end product without themselves being altered at the end of the reactions. That is to say, enzymes are biological catalysts that speed up the rate of biochemical reactions without being altered at the end of the reactions. Now, microbial enzymes are divided into two. We have extracellular enzymes. Extracellular enzymes are generally enzymes that are secreted outside the cell to catalyze the hydrolysis of complex molecules. Example of these enzymes are the digestive enzymes such as proteinases, which break down complex proteins, sucrase, which break down the carbohydrate sugar sucrose, cellulase, which break down the carbohydrate cellulose, amylase, that break down the sugar amylose, caseins, that break down the protein casein, and so on. On the other hand, we have the second subdivision of the enzymes, which are the intracellular enzymes. Intracellular enzymes are a group of enzymes that are secreted within the cell to catalyze catabolic and anabolic reactions inside the microbial cell. Example of these enzymes the permeases, hydrolysis, oxidoreductases, transferases, which we are going to see later in the course of this presentation. Now, if we look at the two categories of enzymes, the examples given, it can easily be deduced that enzyme names end in ASE. So from all these examples, the names of the enzymes ends with ASE. So in the enzyme nomenclature, most of the names end with ASE. Now, regardless of intracellular or extracellular, there are generally 6 classes of enzymes which are defined based on the type of reactions they catalyze. We have the hydrolysis enzymes, which are enzymes that split chemical bonds through the addition of water molecule. We have oxidoreductases enzymes, which are enzymes that catalyze redox reactions. Example, dehydrogenase enzymes. We also have transferase enzymes that catalyze the transfer of functional groups. We also have the lysis enzyme, which split chemical bond without the use of water. That is to say, lysis enzymes act the same as hydrolysis enzymes, only that they do not require the presence of water for their activities. And then we have isomerases enzyme, which are enzymes that catalyzes a rearrangement of atoms within a molecules without changing the number of atoms. That is, they catalyzes isomerization reactions. And the last category of the 6 classes of enzyme is the ligase. Ligase or ligase enzymes are enzymes that forms chemical bond between two group of atoms. Now, having talked about enzymes and their classes briefly, let us now look at the microbial metabolic pathways of energy generations. Let us look at the common metabolic pathways of microorganisms that involves energy generation. Recall, in the previous discussion, we defined metabolic pathways as series of reactions taking place in the body, in which the product of one reaction serve as the substrate of the next reaction. Now, in general, the metabolic pathways of energy generation involve breaking down complex nutrients through the process of catabolism to release energy. These nutrients are of 3 major categories, such as carbohydrates, which are hydrolyzed to glucose, proteins through proteolysis, which are broken down to amino acids, lipids, which are broken down through the process of lipolysis to fatty acids and glycerol. Now, carbohydrates, after the hydrolysis, the glucose is broken down through the process of glycolysis, respiration, or fermentation to release energy. In the case of proteins, they are metabolized through uh, uh, conversion of the amino acids into organic compounds through deamination, decarboxylation, and other reactions, and subsequently those organic compounds are further metabolized through respiration or fermentation to release energy. In the case of lipids, the fatty acid produced oxidized through beta oxidation pathways, while the glycerol are converted to dihydroxyacetone phosphate, which are later metabolized through respiration or fermentation pathway, as we are going to see in the next slides. Now, taking the first category, which is carbohydrate catabolism, to release energy. This process usually takes place through pathways such as glycolysis, respiration, and fermentation. Glycolysis, which is the first steps, is a catabolic pathway in which glucose is broken down through series of enzymatically catalyzed reactions to produce two molecules of pyruvic acids, which are 3 carbon compounds, as well as energy rich compounds in form of ATP and NADH. Glycolysis generally takes place in both aerobic and anaerobic organisms, and it is the first step towards the metabolism of glucose, as we are going to see. Now, there are generally 4 different types of glycolytic pathways. We have the primary glycolytic pathways, which is called the Embden-Meyer-Hofmann's pathway. And then we have the pentose phosphate pathway or hexose monophosphate pathway. Another alternative glycolytic pathway is the Entner-Duodrop pathway. And the last is phosphoketolase pathway. Now, these are the pathways in which glucose is broken down to release energy. For the Embden-Meyer-Hofmann's pathway, glucose is first broken down into 2 molecules of glyceraldehyde phosphate and subsequently into 2 molecules of pyruvic acid with the release of energy. In the case of pentose phosphate pathway, it is an alternative pathway carried out by microorganisms that lack some of the necessary enzymes to follow the normal Embden-Meyer-Hofmann's pathway. In this particular pathway, glucose is converted into phosphogluconate, and then the phosphogluconate is converted into several intermediate compounds, 5 carbon compounds, 4 carbon compounds, 7 carbon compounds, 6 carbon compound, and 3 carbon compounds, and then these intermediate are finally converted into pyruvic acid, which will now join the the normal Embdenmeyer-Hopper's pathway. Now, the next enteroduodrop in enteroduodrop pathway, glucose is also converted into phosphogluconate and then to glyceraldehyde phosphate and pyruvic acid. And then finally, the glyceraldehyde phosphate is converted to pyruvic acid. That is to say, 2 molecules of pyruvic acid are produced together with NADH and ATP. In the next pathway, which is phosphoketolase pathway, glucose is also converted into phosphogluconate, and then the intermediate here are the glyceraldehyde phosphate and acetyl-CoA, which are further metabolized through the normal EMP pathway into pyruvic. The glyceraldehyde is metabolized into pyruvic acid. Now, let us look at the first glycolytic pathway, which is the Embdenmeyer-Hopper's pathway. But before discussing this pathway, there are some points we should try to note. That this being a metabolic pathway, it is characterized by various enzymes which are generally categorized into 5 different categories. We have the first group of enzymes which are called the kinesis enzymes. These enzymes transfer phosphate group from ATP to organic compounds in this particular pathway. Then the second category are the mutasis enzyme. These particular enzymes rearrange and move phosphate, high energy phosphate group within a molecule, as we are going to see. And then we have isomerase enzyme, which, as we already discussed, is an enzyme that catalyzes the isomerization of molecules. And then, of course, we have the dehydrogenase enzyme. This enzyme catalyzes redox reaction. It oxidizes an organic compound through dehydrogenation, removal of hydrogen and transferring the hydrogen to coenzyme NAD to produce NADH. And the last category of enzyme we are going to encounter is the enolase enzyme. This particular enzyme carries out dehydration reaction. It removes molecules of water from the molecules. Now, all these enzymes are named based on 2 criteria. The first criteria is based on the type of reactions they catalyze, and the second criteria is based on the substrate they act upon, like the kinesis enzyme that transfer phosphate group from ATP to glucose is called hexose kinase. Hexose based on 6 carbon sugar, and then kinesis based on the function of the enzyme, as we are going to see. Now let us look at the Embdenmeyer-Hopper's pathway in detail. Embdenmeyer-Hopper's pathway is generally divided into 2 stages. The first stage is the energy investment stage, while the second stage is the energy producing stage. So in the first stage, energy is invested. That is, energy from ATP is utilized to prepare the reactant for the reaction, while in the second phase, energy is being produced from the organic compounds. Now let's look at the first phase. In the first phase, as we already said, this pathway starts with glucose. We have glucose with carbon atom 1 to 6. Now, in glucose being intact, for glucose to start this particular reactions, it has to be energized. That means it has to be acted upon by a kinase enzyme. So we have a kinase enzyme, which is a hexose kinase that carry out phosphorylation of glucose. It transfers high energy phosphate group to glucose, energizing the glucose molecules. So by transferring the energy to glucose, it produces an unstable product called glucose 1-phosphate. Immediately this product is formed, it is rearranged by transferring the phosphate group to carbon atom number 6 through the action of the phosphoglucomatase. As we said, the enzymes are named based on the substrate they act upon. The mutase transfers the phosphate group, as we explained earlier, and then the name phosphogluco is from glucose phosphate. Now, once glucose phosphate is produced, it is isomerizes into fructose 6-phosphate by glucose 6-phosphate isomerase. You see the enzyme is named based on the substrate, glucose 6-phosphate, and then based on the function it carries, which is isomerase. Now, fructose 6-phosphate is a kind of stable product. In order to continue to the next stage of the reaction, it has to be energized again. So in this case, we have another kinase enzyme which is called fructose kinase, based on the name of the substrate it acts upon, fructose, and kinase based on the function which transfer high energy phosphate group from ATP to fructose 6-phosphate to produce fructose 1,6-diphosphate. This product is of high energy content, and therefore that makes it become unstable. And in the presence of an enzyme aldolase, this product is cleaved into 2,3 carbon compounds, which the first of which is dihydroxyacetane phosphate, and the second one is glyceraldehyde phosphate. These two compounds are isomers of each other. They can be converted to one another by an enzyme called triose phosphate isomerase or glyceraldehyde phosphate isomerase, if we want to name it based on the substrate it acts upon. So generally, the dihydroxy, there is no enzyme that further act on dihydroxyacetone, and therefore it has to be isomerized to glyceraldehyde phosphate. So at the end of this stage, we are going to have 2 molecules of glyceraldehyde phosphate that will move into the energy producing phase. Now, in the energy producing phase, the glyceraldehyde phosphate is first oxidized by the dehydrogenase enzyme, which is known as glyceraldehyde dehydrogenase. This particular enzyme does 2 functions. Number 1, it dehydrogenates glyceraldehyde phosphate by transferring the hydrogen group from glyceraldehyde phosphate to the coenzyme NAD to produce NADH, and secondly, it transfer organic phosphate to replace the position of the hydrogen that has been transferred to NAD. Now with this, it leads to the production of 1,3-diphosphoglyceric acid, which is of high energy content, having 2 phosphate groups. And therefore, in the next step, the dihydroxyacetone as the 1,3-diphosphoglyceric acid undergoes substrate level phosphorylation under the action of the enzyme phosphoglycerate kinase, which transfer one of the phosphate group to ADP to produce the first ATP. Now, by doing this, 1,3-diphosphoglyceric acid will now become 3-phosphoglyceric acid. This 3-phosphoglyceric acid is an unstable product, and therefore it is being rearranged by the mutase enzyme, which is the phosphoglyceromutase, which try to balance the phosphate group by transferring the phosphate group to carbon atom number 2 to enable the binding site of the next enzyme. Now, this particular product is somewhat stable, and therefore it has to be dehydrated to make it unstable. A molecule of water is being removed by an enzyme called enolase, as we already explained. So by removing a molecule of water, the compound, the 2-phosphoglyceric acid becomes phosphoenol pyruvic acid, which is an unstable product, and therefore it easily liberates the high energy phosphate group, which is being transferred to ADP to produce ATP through another substrate level phosphorylation under the action of the enzyme pyruvate kinase. And the final product that will be formed is pyruvic acid, which is the end product of glycolysis. So at the end of glycolysis, the two dihydroxyacetone phosphate and glyceraldehyde phosphate are converted into pyruvic acid. 2 ATP are produced, and then 1 NADH is produced from each of them. So in total, 2 ATPs are harvested during the energy investment phase, while in the energy producing phase, 4 ATP are produced, 2 ATP from glyceraldehyde phosphate and 2 from dihydroxyacetone phosphate, and then 2 NADH are also produced, 1 from glyceraldehyde phosphate and 1 from dihydroxyacetone phosphate. So the net ATP, that is the net energy that is produced, or let's say the final product will now be 2 molecules of pyruvic acid plus the net ATP, which is now 2 ATP. 4 ATP minus the 2 ATP invested initially. So the net ATP is 2 ATP plus 2 NADH. These are the final product of glycolysis. Now, after glycolysis, in aerobic organisms that can utilize oxygen, usually they further oxidize the pyruvic acid produced into carbon dioxide and water to produce large amount of energy in a cycle which is called the tricarboxylic acid cycle or citric acid cycle or Krebs cycle. Now, this particular cycle is a cyclic enzymatically catalyzed pathway in which the pyruvate produced in glycolysis is oxidized first into acetyl-CoA, and subsequently the acetyl-CoA is oxidized by combining with oxaloacetic acid to produce citric acid, and then oxidized to release carbon dioxide, ATP, and energy-rich compound in form of NADH and FADH2, and finally regenerate the oxaloacetic acid that has been used initially. This particular pathway usually take place in the mitochondrial matrix of eukaryotic cells and cytoplasm of prokaryotic cells. The Krebs cycle serves as the center of cellular metabolism, which link various metabolic pathways, and it plays a crucial role in energy production, as we are going to see. Now let us look at the pathway in detail. The first step in the pathway, which is the preparatory stage, is that the pyruvate from glycolysis is first decarboxylated through dehydrogenation using pyruvate dehydrogenase enzyme complex that use coenzyme A that attach coenzyme A in the place of decarboxylated carbon. So what happens is that the carbon dioxide is removed through decarboxylation, and then acetyl-CoA is placed in place of the carbon dioxide, replaces the carbon dioxide, CoH replaces the carbon dioxide, and the final product is acetyl plus CoH, which is called the acetyl CoA, under the action of the enzyme pyruvate dehydrogenase. Now the next step of the reaction is where the acetyl-CoA enters into Krebs cycle. In order to enter into Krebs cycle, the acetyl-CoA is condensed, is coupled with an organic acid present in the cytoplasm of most microorganisms, which is oxaloacetic acid, which is a 4-carbon atom, to form 6 carbon compounds. In the process, the CoH is liberated under the action of the enzyme citrate synthetase to produce the 6 carbon compound, which is called citrate or citric acid. Now, in the next step of the reaction, involves isomerization of the citric acid to aconitate. The essence of this isomerization is to change the position of the hydroxyl group to enable the action of the next enzyme. So in order to do this, aconitase dehydrate citrate first to become cis-aconitate, and then rehydrate it back. So during the rehydration, the position of the OH group is changed from before it was on this carbon atom number 3, and now on this carbon atom number 5. So the final product that is produced through this rehydration is called isocitric acid, which is an isomer of the citric acid that was produced initially. Now the isostric acid is acted upon by the next enzyme, which is the isostrate dehydrogenase enzyme. This enzyme, just like we have in glycolysis, dehydrogenase enzymes are enzymes that remove hydrogen and transfer to NAD to form NADH. So in essence, they oxidize an organic compound by removing hydrogen and using it to reduce NAD to NADH. So in the process, decarboxylation takes place. So one of the carbon atom from the 6 carbon has been removed as carbon dioxide. So what we have now is 5 carbon compound, which is alpha ketoglutaric acid or alpha ketoglutarate. Now, after the formation of alpha ketoglutarate, the next is to oxidize the remaining carbon compound. Let us recall that 2 carbon compounds comes from glycolysis, enters the Krebs cycle. So these 2 carbon compounds are oxidized, oxidized to produce NADH, and carbon dioxide is liberated in these 2 stages. So in this stage, the enzymes that act upon this, which is named based on the substrate alpha ketoglutarate, is alpha ketoglutarate dehydrogenase, which uses CoAsh. It decarboxylates the alpha ketoglutarate and attach and transfer CoAsh to the product in place of the carbon dioxide, forming the next product, which is the succinyl CoA. Now this succinyl CoA is converted into succinic acid or succinate by succinyl CoA synthetase enzyme, which is an enzyme that hydrates the product, and in the process the CoAsh is liberated, and the energy that is produced is used to convert GDP, guanosine diphosphate, into guanosine triphosphate, which is equivalent to 1 ATP molecule. So this is the first direct energy that is produced in Krebs cycle. Now the succinate, after this stage, the next steps is to regenerate the oxaloacetic acid. In order to regenerate the oxaloacetic acid, the succinic acid that is produced here is acted upon by an enzyme succinate dehydrogenase to make it unstable, because it is already now a stable product. So in order to proceed to the next reaction, it has to be unstabilized by becoming unsaturated using the coenzyme FAD, which receives the hydrogen to become FADH2, and the resulting product is the fumarate or fumaric acid. Now the fumarate is now rehydrated, is hydrated by an enzyme called fumarase or fumarate hydratase enzyme, and the water which is transferred as hydroxyl and hydrogen atoms break the double bond, and the resulting product become malate or malic acid. Now the malic acid is finally converted into oxaloacetic acid through dehydrogenation to generate the ketone group in oxaloacetic acid. So during the dehydrogenation, the hydrogen is being transferred to NAD to produce NADH. So this is citric acid cycle. It consists of three different phases. We have the preparatory stage, in which the pyruvate is first converted into acetyl-CoA, and then we have the second stage, in which the acetyl-CoA is being oxidized. The carbon compounds are being oxidized and being released as carbon dioxide. And then the next step is the regeneration of oxaloacetic acid. So these are the three different stages of Krebs cycle. So at the end of Krebs cycle, we have 3 carbon dioxide being produced, 1 carbon dioxide at the preparatory stage, and 2 carbon dioxide that are produced here in the during the phase 2 dehydrogenation reaction. In addition, NADH is liberated. 1 NADH is liberated at the preparatory stage, and then 3 NADH are produced during the main cycle. The first one in the first and second one during the first 2 dehydrogenations, while the last NADH is produced in the last dehydrogenation reaction. In addition, 1 FADH2 is produced during the process. So for the 2 pyruvic acid compounds, 6 NADH is produced, 2 FADH are produced, 2 ATPs are produced from this combustion, and then 6 carbon dioxide are produced. Remember, glucose has 6 carbon atoms. So 6 carbon dioxide, after complete oxidation, we have 6 carbon dioxide being produced. So now let us look at the summary of the energy yield from the 2 oxidation steps, glycolysis and Krebs cycle. In glycolysis, glucose, we have the N in the glucose is converted into 2 molecules of pyruvic acid, during which 2 molecules of ATP are utilized, 2 NADH are produced, and 4 ATP are produced, making a net ATP of 2 ATP and 2 NADH being produced during glycolysis. And then in the preparatory stage of Krebs cycle, these 2 pyruvic acid are decarboxylated, yielding 2 carbon dioxide molecule, and then 2 NADH are produced in the process, and the pyruvic acid are converted into acetyl-CoA that enters Krebs cycle. So in the Krebs cycle, the acetyl-CoA is utilized to produce 2 molecules of carbon dioxide for each acetyl-CoA, that is 4 molecules of carbon dioxide, and then we have 2 NADH, 1 from each dehydrogenation step, as we already see. So total NADH will be 6 NADH, 2 ATPs are produced, and 2 FADH are produced. So this is a summary of the energy compounds that are produced from the process. Now let us look at the net ATP produced based on these processes. So in glycolysis, we have 2 NADH produced and 2 ATP produced. Now these 2 NADH usually moves into electron transport chain and chemiosmosis, where they are oxidized to produce ATP. So during the preparatory stage of of citric acid cycle, another 2 NADH are produced, and then more NADH are produced during Krebs cycle. So in total, 10 NADH are produced, 2 FADH2 are produced from both glycolysis and Krebs cycle. Now these NADH and FADH2 are oxidized back to NAD and FAD in order for the cycle to continue. And during this oxidation process, 3 molecules of ATP are produced from each NADH, making 30 ATP from the 10 NADH, and then 2 molecules of ATP are produced from each FADH2, making 4 ATP being produced from the 2 FADH2, and therefore we have a total of 34 ATP being produced from the 10 NADH and 2 FADH2. So adding together with the 2 ATP produced from glycolysis and 2 ATP produced in Krebs cycle, we have a total of 38 net ATP produced from both glycolysis and Krebs cycle. Now, let us look at the alternative glycolytic pathways apart from the primary glycolytic pathway. The first alternative glycolytic pathway is the pentose phosphate pathway, as we already mentioned earlier. Now, the pentose phosphate pathway, which is also called the hexose monophosphate pathway, is a metabolic pathway in which glucose is metabolized into a variety of products, including ribose 5-phosphate, erythrose 4-phosphate, and NADPH. These intermediates are used for many anabolic processes such as lipid and nucleic acid synthesis, as well as glucose synthesis from carbon dioxide in photosynthesis, and some amino acids. Now, the pathway occurs in two ways: the oxidative phase and the non-oxidative phase. In the oxidative phase, glucose 6-phosphate is converted into NADPH and ribose 5-phosphate, while in the non-oxidative phase, combustion of multiple sugars, including ribose 5-phosphate, erythrose 4-phosphate, the intermediate produced into array of sugars and related products take place. Now let us look at the pathway in detail. Now the first step in the pathway is similar to that of primary glycolytic pathway, which is the Embden-Meyer-Hoffernes pathway, in which glucose is converted into glucose 6-phosphate, is phosphorylated by hexose kinase enzyme. Now, for pentose phosphate pathway, usually 3 glucose molecules, the pathway takes 3 glucose molecules at a time. So 3 glucose molecules are oxidized are phosphorylated using 3 ATP to give 3 glucose 6-phosphate molecule. Now, in the next step, the glucose 6-phosphate is dehydrogenated, unlike in primary glycolytic pathway where glucose 6-phosphate is isomerized to fructose. Here it is dehydrogenated. The microorganisms mostly doesn't have the enzymes for the other pathway, therefore they use an enzyme glucose 6-phosphate dehydrogenase to dehydrogenase glucose 6-phosphate to 6-phosphogluconate. The 6-phosphogluconate is further dehydrogenated and decarboxylated by 6-phosphogluconate dehydrogenase to produce NADPH and carbon dioxide, and the decarboxylation leads to the production of ribulose 5-phosphate. Now, this ribulose 5-phosphate is the central sugar in pentose phosphate pathway, which is converted into several intermediates, as we are going to see. First, the ribulose 5-phosphate is isomerized by ribulose 5-phosphate isomerase into ribose 5-phosphate. It is also isomerized using epimerism by epimerase enzyme to xylulose 5-phosphate. Now, the xylulose 5-phosphate and ribose 5-phosphate recombine together and during the recombination, ketone group is being transferred from the xylulose 5-phosphate to ribose 5-phosphate, and therefore the xylulose 5-phosphate become 3-carbon compound glyceraldehyde phosphate, while the ribose 5-phosphate becomes pseudoheptylase 7-phosphate under the action of the transketolase enzyme. Now, these two products that are produced recombine again under transaldolase, in which the aldehyde group is being transferred from pseudoheptylase to the glyceraldehyde phosphate to become erythrose 4-phosphate, while the pseudoheptylase become fructose 6-phosphate. Now, in the next reaction, the other molecule of xylulose 5-phosphate combines with erythrose 4-phosphate to produce fructose 6-phosphate and glyceraldehyde 3-phosphate under the action of the enzyme transketolase, and hence the glyceraldehyde phosphate is further converted into will now join the normal glycolytic pathway and be converted into pyruvic acid, or sometimes it is converted into glucose to regenerate glucose. While the fructose 6-phosphate is converted back to glucose during glucose synthesis. So that is why this pathway is more of synthetic than energy producing pathway. Now, in summary, 3-glucose added to 3 molecules of water plus 3 ATP plus 6 NADP to produce pyruvate plus 2 fructose phosphate plus 6 NADPH plus 3 carbon dioxide molecules in pentose phosphate pathway. Now, having looked at the pentose phosphate pathway, let us look at the another alternative glycolytic pathway, which is the Entner duodrop pathway. In Entner duodrop pathway, as I said, it's an alternative glycolytic pathway, and this pathway is commonly used by gram-negatives, which are the soil bacteria mostly, Rhizobium, Pseudomonas, Agrobacterium, and so on. It is not usually carried out by gram-positive organisms. And in NTNA-drop pathway, glucose is oxidized to pyruvic acid and glyceraldehyde 3-phosphate. And in the process, ATP and NADPH are produced, which are used for biosynthetic process. Now let's look at the pathway in detail. It starts with conversion of glucose to glucose 6-phosphate, like all the other glycolytic pathways, after which glucose 6-phosphate dehydrogenase is produced, which convert glucose 6-phosphate to 6-phosphogluconate, just like in pentose phosphate pathway. Now the subsequent reaction is what differentiates pentose phosphate pathway and NTNA-drop pathway. In NTNA-drop pathway, an enzyme called 6-phosphogluconate dehydratase is produced, which removes water molecules from 6-phosphogluconate, leading to the formation of 2-keto-3-deoxy 6-phosphogluconate KDPG compound. Now this compound is unstable, and therefore it is cleaved by KDPG aldolase enzyme into two molecules, one pyruvic acid and glyceraldehyde phosphate. The glyceraldehyde phosphate can be converted into pyruvate through normal glycolytic pathway, and hence, in summary, in this pathway, glucose plus ATP plus NADP produce 2 molecules of pyruvate plus NADPH plus NADH plus 2 ATP molecules. Now, the last alternative glycolytic pathway is the phosphoketolase pathway. Phosphoketolase pathway is also known as the heterolactic pathway. It's a metabolic pathway that occurs in various bacteria, yeast, and archaea, and it involves the breaking down of carbohydrates, particularly the pentose sugars, into different metabolic intermediates, such as the acetyl phosphate, acetyl-CoA, erythrospore phosphate, which are used for various synthetic processes. So the pathway consists of 3 key stages, which we are going to see. In the first step, glucose is phosphorylated to glucose 6-phosphate like other glycolytic pathway, and then glucose 6-phosphate is dehydrogenated to 6-phosphogluconate like the pentose phosphate pathway and NTNA-drop pathway. Then next, the 6-phosphogluconate is dehydrogenated to ribulose 5-phosphate, just like in pentose phosphate pathway. However, in this pathway, ribulose 5-phosphate epimerase convert ribulose 5-phosphate into xylulose 5-phosphate, and then the first steps that differentiated with other pathways start, in which the xylulose 5-phosphate is converted into glyceraldehyde phosphate and acetyl phosphate by phosphoketolase enzyme. Next, the glyceraldehyde phosphate is converted into pyruvate or erythrospore phosphate, which can be utilized for synthesis of nucleic nucleotide and aromatic amino acid. In the final stage, the acetyl phosphate is converted into acetyl-CoA by acetate kinase. So in summary, in this pathway, glucose plus ATP plus 2 NADP produce glyceraldehyde phosphate plus NADPH plus acetyl-CoA, or pyruvate plus NADPH plus acetyl-CoA. Now, having discussed the 4 different pathways of carbohydrate catabolism, let us now look at the pathways of lipid catabolism. In lipid catabolism, lipids are first broken down into fatty acid and glycerol, which is converted to acetyl-CoA that can enter into Krebs cycle. So in essence, let's take an example of lipid, which is triglyceride. Triglyceride is a lipid that contains one molecule of glycerol bonded to 3 molecules of fatty acid. Now, triglyceride is first broken down into glycerol and fatty acid by the lipase enzyme, which utilize water molecules to produce 1 molecule of glycerol and release 3 different molecules of fatty acid. Now, in the next step of the process, the glycerol isomerizes to dihydroxyacetone phosphate, which will now join glycolysis to produce acetyl-CoA that can enter Krebs cycle for complete oxidation. While the fatty acids become oxidized through beta oxidation pathway to produce several molecules of acetyl-CoA that can enter into Krebs cycle. Now, let us look at the beta oxidation pathway briefly. In beta oxidation pathway, each fatty acid combines with CoH in the presence of adenosine monophosphate to produce fatty acyl-CoA, and in the process a water molecule is liberated plus the adenosine monophosphate. Now the fatty acyl-CoA is now the first product that enters into beta oxidation pathway. In the first step of the beta oxidation pathway, the fatty acyl-CoA is acted upon by acyl-CoA dehydrogenase enzyme that uses FAD to dehydrogenate fatty acyl-CoA into enoyl-CoA to produce an enoyl-CoA, creating double bond. Remember, FAD usually creates double bonds in organic compounds, unlike NAD that usually decarboxylate. Now, in the next step, the enoyl-CoA is acted upon by enoyl-CoA hydratase, which hydrate the unsaturated enoyl-CoA to become beta hydroxyacyl-CoA. Now, the beta hydroxyacyl-CoA is acted upon by hydroxyacyl-CoA dehydrogenase that use NAD to dehydrogenate beta hydroxyacyl-CoA to produce NADH and beta ketoacyl-CoA. Now, beta ketoacyl-CoA is acted upon by thiolase, which cleaves the acetyl-CoA group from the beta ketoacyl-CoA, and once the acetyl group is removed, the final product become during this process, CoH is used to replace the place of the acetyl-CoA, and the final product become fatty acyl-CoA. The fatty acyl-CoA will now repeat. It will now repeat the process, and the cycle continue. Each time 2 carbon units are being removed in form of acetyl-CoA until the complete fatty acid is oxidized into acetyl-CoA that can join Krebs cycle. Now, having looked at the catabolism of lipids, let us now look at the protein catabolism. In protein catabolism, proteins are first converted into peptide and then into amino acids, which are further converted into organic acids that can join normal and the Meyer-Hofmann's pathway. Now let's look at the stages. The first stage, the complex proteins are first broken down into peptide by the proteases enzymes, and then subsequently the peptides are broken down into individual amino acid by the peptidases enzymes, and subsequently the amino acids are converted into various organic acids through several types of reactions such as deamination, removal of amino group, transamination, transfer of the amino acid group to organic acid, decarboxylation, removal of the COOH group in the amino acid, or dehydrogenation, which is removal of hydrogen. For example, this is an example of deamination. We have an amino acid being acted upon by the aminase enzyme and to remove the amino group, leading to liberation of ammonia and formation of an organic acid that can join the EMP pathway. And the second example, we have transamination reaction, in which we have the amino acid alanine containing an amino group, in which the amino group in alanine is being transferred to alpha ketoglutarate, which is an organic acid. So by transaminase enzyme, leading to the formation of pyruvate. The alanine now becomes pyruvate, while the alpha ketoglutarate becomes glutamic acid. And the next example is the decarboxylation of amino acid, in which an amino acid is decarboxylated to give organic acid plus carbon dioxide. So in essence, amino acids have to be converted into organic acids that can join normal glycolytic pathway for oxidation. Now, in summary, the summary of carbohydrate, protein, and lipid metabolism. In summary, proteins have to be converted into peptides, into amino acids, and then the amino acids become converted into various organic acids that can join the normal glycolytic pathway. This is the central glycolytic pathway, which is the Meyer-Hofmann's pathway, in which carbohydrates are converted into sugars to glucose, which are oxidized through glycolysis to Krebs cycle and electron transportation to produce energy. In the case of lipids, they are being broken down into glycerol and fatty acids. Glycerol become isomerized to glyceraldehyde phosphate and to join the glycolytic pathway, while fatty acids are converted into acetyl-CoA through beta oxidation pathway to join the normal glycolytic pathway. So in both, this is the summary of the nutrient microbial nutrient catabolism of energy production. Now, what we have seen previously is aerobic respiration, in which glucose is metabolized completely into carbon dioxide and water to produce large amount of energy. In the case of anaerobic microorganisms, in the absence of oxygen, the NAD, the FADH cannot be regenerated, and therefore Krebs cycle cannot take place. Therefore, these organisms utilizes fermentation pathway, which involve partial anaerobic oxidation of food to produce a small amount of energy. Fermentation is a metabolic process in which sugars, amino acid, and other organic compounds are metabolized in the absence of oxygen to produce small amount of energy and other usable products. So generally, ATP production in fermentation begins and ends with glycolysis. So in order to sustain ATP generation by glycolysis, the NADH produced must be oxidized back to NAD. That is to say, the essence of fermentation is to regenerate the NAD, is to convert the NADH back to NAD so that glycolysis can continue to produce energy. In the process, organic compounds are produced. In glycolysis, NAD is converted to NADH, while in fermentation, NADH is regenerated. NADH is converted, is oxidized back to NAD. So the starting point of fermentation is glucose or any other fermentable carbohydrate. Therefore, proteins and lipids have to be converted into glucose via gluconeogenesis, production of glucose from other sources, in order to join fermentation pathways. So when they are converted into glucose, the glucose will now pass through glycolysis, as we explained, to produce glyceraldehyde phosphate, and then next, the glyceraldehyde phosphate is oxidized to pyruvic acid. In the process, ATP is produced and NAD is converted into NADH. Now, once this is produced, the pyruvate or pyruvic acid produced will now proceed to fermentation pathways instead of Krebs cycle in the absence of oxygen, because the coenzymes in Krebs cycle cannot be generated in the absence of electron transport chain that can oxidize them back to their oxidized forms. So depending on various microorganisms, the pyruvic acid is converted to several fermentation products. For example, Streptococcus species usually oxidize NADH, regenerates their NAD by oxidizing NADH to NAD through lactic acid fermentation. So in lactic acid fermentation, the NADH produced here is used to reduce the pyruvic acid to produce lactic acid. So in this process, the NAD has been regenerated, which will go back and then the normal glycolysis can continue. In the case of other organisms like yeast, yeast regenerates their NAD through alcohol fermentation pathway. So in yeast, the pyruvate is first decarboxylated by pyruvate decarboxylase to produce carbon dioxide and acetaldehyde, which is reduced using the NADH to ethyl alcohol or ethanol. In the process, the NAD is regenerated, which can go back to normal glycolysis. So here are examples of various other fermentation products that are produced by various microorganisms. Like we give examples, Streptococcus carried out lactic acid fermentation. In the process, NADH is oxidized to NAD. We have Saccharomyces cerevisiae, which are the yeast species that carry out alcohol fermentation to produce ethanol and carbon dioxide. We have Propionibacterium that produce propionic acid, acetic acid, carbon dioxide, and hydrogen gas. We have Clostridium that produce butyric acid, and so on. So all these ones are various fermentation products that are produced by microorganisms, various microorganisms. So with this, we have come to the end of the first part of introduction to microbial metabolism. So I wish to say thank you for listening, wishing you best of luck. Thank you.